Hello guys, in this video we are going to study about the structure of the sperm and spermatogenesis. The sperm contains a head, a neck, a middle piece and a principal piece or a tail. The head contains a nucleus and an acrosomal cap. The nucleus contains 23 highly condensed chromatins which make it extremely resistant to stresses. The acrosome contains enzymes which help in the penetration of the sperm into the ovum during the fertilization. The neck contains a proximal centriole and a basal body. The middle piece contains an axial filament which is surrounded by mitochondrial sheath. The axial filament extends from the middle piece into the tail. The axial filament is actually composed of various fibrils. The central pair of fibrils is surrounded by 9 pairs or doublets in a circle. Immediately outside the fibrils, there is a fibrous sheath. In addition to the doublets, the axial filament contains 9 petal-shaped fibrils, one such fibril lying just outside each doublet. Spermatogenesis. In a human male, the formation of spermatozoa takes place only during the reproductive period, which begins at the age of puberty, that is 12 to 16 years, and it continues even through old age. Spermatozoa are formed in the walls of seminiferous tubules of the testis. If we look closely at these seminiferous tubules under a microscope, we will find cells of different shapes and sizes. These cells represent various stages of spermatogenesis. The various cell stages in spermatogenesis are as follows. The spermatogonia type A divide mitotically to give rise to more spermatogonia of type A and also to spermatogonia of type B. The spermatogonia type B undergo mitosis to form primary spermatocyte. These primary spermatocytes are the cells that undergo first meiosis and form secondary spermatocytes. The secondary spermatocytes undergo second meiotic division to form spermatids. These spermatids then transform into sperm. This entire process of formation of sperm from spermatogonium is called spermatogenesis, while the process by which a spermatid becomes a spermatozoa is called spermiogenesis. The spermatids are more or less circular cells. They contain a nucleus, a Golgi apparatus, centriole, and mitochondria. The nucleus forms the head. The Golgi apparatus is transformed into the acrosomal cap. The centriole divides to form two parts. One centriole becomes spherical and comes to lie in the neck. The other centriole becomes ring-shaped and forms the annulus. The axial filament appears to grow out of centrioles. This axial filament lies in the middle piece where it is surrounded by mitochondrial sheath and extends into the tail. Most of the cytoplasm of spermatid is shed, but the cell membrane persists as a covering for the spermatozoa. When first formed in seminiferous tubules, spermatozoa are immature. A current of fluid in seminiferous tubules carries spermatozoa from testis to epididymis. Here they are stored and undergo maturation. Spermatozoa acquire some motility after maturation but become fully motile only after ejaculation when they get mixed up with the secretion of prostate glands and seminal vesicles. The ability to fertilize an ovum is acquired only in the female genital tract. This final step in maturation of sperm is called capacitation. In the process of capacitation, the glycoprotein coat and seminal proteins lying over the surface of spermatozoa are altered.